Hello and welcome to IB Biology Notes with Dr. Kimo. This is A1.2 Nucleic Acids. And these are the contents of the current subchapter. DNA carries the information of life. Genetic material stores hereditary information. That's information that's inherited from one generation to the next. All living organisms use DNA as their genetic material, and DNA stands for deoxyribonucleic acid. There's also another form of nucleic acid, RNA, ribonucleic acid. Nucleic acids are polymers, which are made up of monomers called nucleotides. Mono means one, and poly means many. Nucleotides have three components. 1. A phosphate group, which is acidic and negatively charged. 2. A sugar with 5 carbon atoms, that's a pentose sugar. In DNA, the pentose is called deoxyribose, and in RNA, it is called ribose. 3. A nitrogenous base with 1 or 2 rings. DNA and RNA have a sugar-phosphate backbone. Nucleotides bind to each other through covalent phosphodiester bonds. The sugars and the phosphate groups of the nucleotides form the backbone of the DNA and RNA strand. Here below you can see a computer-generated ball and stick model of a double-stranded DNA molecule, and the yellow dotted line follows one of the two backbones. DNA has 4 different types of bases: adenine, thymine, guanine, and cytosine. A, T, G, and C. A and G are purines. They have 2 rings. T and C are pyrimidines. They have 1 ring. And these bases are like the letters in the book of life. They are organized in triplets. So it's a bit like a 3-letter word, triplet. With 4 letters, there can be 4 times 4 times 4. That's 64 different triplets. So 64 different 3-letter words. We will learn more about triplets later. RNA also has 4 different types of bases. Instead of T, there's U, uracil. Uracil is a pyrimidine. RNA is a polymer consisting of an unbranched chain of nucleotides. Each nucleotide is therefore a monomer and carries a nitrogenous base, either adenine, uracil, guanine, or cytosine. A new nucleotide adds to a growing RNA molecule by a condensation reaction, where 2 hydroxyl groups are used. One hydroxyl group is removed and combined with a hydrogen from another hydroxyl group, whereby water is released. And that's why it's called a condensation reaction. DNA is a double helix. It has 2 unbranched nucleotide chains. These chains or strands are connected by hydrogen bonds between the nitrogenous bases A, T, G, and C. A forms hydrogen bonds with T. They form a complementary base pair. G forms hydrogen bonds with C. They form a complementary base pair. The 2 strands run opposite to each other. They are anti-parallel. And their shape is helical. They form a double helix. DNA and RNA differ in 3 important ways. The number of strands. DNA is double-stranded, whereas RNA is single-stranded. The bases. DNA has ATGC, whereas RNA has AUGC. And the sugar. DNA has deoxyribose, whereas RNA has ribose. DNA replication. All DNA in a cell is copied or replicated before the cell divides. During DNA replication, the double strand, which is blue here, opens up and new nucleotides are added to form two complementary strands, which are green here. And that happens through the process of complementary base pairing. DNA transcription. Complementary base pairing also allows the information in DNA to be copied or transcribed into RNA. The information in RNA is then translated into protein. So we go from DNA to RNA to protein. The first step when information is transferred from DNA to RNA is called transcription. The second step, when information is transferred from RNA to protein, is called translation. But you will learn much more about this later on. Information storage in DNA. In computers, information comes in bits. There are two types of bits, 0 and 1. The bits come in groups of 8, also known as octets. And in computers, the octets are also called bytes. In DNA, information also comes in bits. There are 4 types of bits, A, T, G, and C. And the bits come in groups of 3, also known as triplets. And more about triplets in a later chapter. Information storage in DNA. In DNA, any sequence of bases is possible. With 4 different bases, there are 4 to the power of 2 different 2-base sequences. AA, AT, AG, AC, and so on. 16 possibilities. For a 3-base sequence, there are 4 to the power of 3 possibilities. That's 64. For a 4-base sequence, there are 4 to the power of 4 possibilities. That's already 256. Now for a 50-base sequence, the number of possibilities is 4 to the power of 50, and that is a huge number. The average human gene has more than 53,000 bases. This means that enormous amounts of information can be stored efficiently in DNA in a very limited space inside the nucleus of a cell. The genetic code is the same in all living things. In order to carry out its functions, a cell needs to build many different proteins with specific shapes and functions. Proteins consist of amino acids, and the DNA code contains information about which amino acids and in which order a protein should consist of. DNA can contain 4 to the power of 3, that's 64 different base triplets. Each triplet has a meaning. Most of them correspond to a specific amino acid, whereas some triplets tell the cell where the code for a protein starts or ends. Amazingly, this code is identical in all living organisms and even in viruses, with a few small exceptions. This is another piece of evidence of universal common ancestry for all life on Earth. Now we are moving into higher level territory, the directionality of RNA and DNA. RNA and DNA strands have a 5' end and a 3' end. The carbon atoms in ribose and deoxyribose are numbered 1 to 5. And when these monomers, the nucleotides, form polymers, strands, the strands are formed by bonds between the 3' carbon of one nucleotide and the phosphate group which is attached to the 5' carbon of another nucleotide. Complementary DNA strands run in opposite direction. They are anti-parallel. And if you look at the strand in the middle of the screen and compare it with the strand on the right side of the screen, you will see that the one on the right side is sort of upside down. It's anti-parallel. Replication, transcription, and translation occur in a 5' to 3' direction. On the left, we have replication, when DNA copies itself. The new DNA strand, the blue strand, is assembled in a 5' to 3' direction. In the middle, you have transcription, when information in DNA is transferred to RNA. The orange RNA strand is also assembled in a 5' to 3' direction. On the right side, you have translation, when the information in RNA is transferred to protein. The ribosome, which is red in this picture, moves along the RNA strand in a 5' to 3' direction. Purine to pyrimidine bonding as a component of DNA helix stability. Each DNA base pair consists of a purine and a pyrimidine, either adenine and thymine or cytosine and guanine. Purines, like adenine and guanine, have 2 rings, and pyrimidines, like thymine and cytosine, have 1 ring. Each complementary base pair has 1 purine and 1 pyrimidine, and therefore there are always 3 rings in a pair, and the distance between the backbones of 2 complementary strands is always the same. This contributes to the stability of the double helix. DNA and histone proteins form nucleosomes. A human cell contains about 2 meters of DNA. The DNA fits into a cell because it's densely packed in a highly organized way. The smallest unit of organization is the nucleosome. A nucleosome consists of around 150 base pairs wrapped around 8 histone proteins, with the 9th one on the outside. The image in the middle of the screen shows this. The image on the right side of the screen shows the same thing, but that is a structural computer model of a nucleosome. Let's look at 2 discoveries. First, Hershey and Chase, who showed that DNA is the genetic material. In the early 1940s, the nature of the genetic or hereditary material was not known. There were 2 main candidates, proteins and nucleic acids. It was known that bacteriophage-infected bacteria produce phage proteins. Bacteriophages are viruses that infect bacteria. They consist of protein and DNA. Infection by bacteriophages requires that the phage genetic material enters the bacteria. And Alfred Hershey and Martha Chase showed that when bacteriophages infect bacteria, Their DNA enters the bacteria while the protein does not. This is how they did it. Hershey and Chase cultivated one batch of phages in the presence of radioactively labeled sulfur 32S sulfur 32, left column. Protein contains sulfur, so the phage's protein capsule became labeled. They also cultivated another batch of phages in the presence of radioactively labeled phosphorus 40P phosphorus 40, right column. DNA contains phosphorus, so the phage's DNA became labeled. Then they added the 32S phages to one bacterial culture and the 40P phages to another bacterial culture. The phages infected the bacteria, and that is step 1 in this illustration. The cultures were then put in a blender to detach any external virus parts from the cell. That's step 2. And it meant that any virus parts which had not entered the cell were separated from the cell. After some time, the mix was centrifuged. That's step 3. And on the left you can see a centrifuge in a lab. After centrifugation, the bacteria ended up in the solid pellet at the bottom of the tube, whereas any free-floating molecules stayed in the liquid supernatant. In the experiment with sulfur 32, there was little radioactivity in the pellet, that's in the cells. In the experiment with phosphorus 40, there was a lot of radioactivity in the pellet, that's in the cells. The conclusion was that the phosphorus 40 containing DNA had been inserted by the phages into the bacteria, which indicates that DNA is the genetic material. And now to another discovery. In the late 1940s, Erwin Chargaff showed that in DNA, the amounts of adenine and thymine are equal. He also showed that the amounts of guanine and cytosine are equal. The number of cytosine guanine can vary from that of adenine thymine. He tested several species, human, octopus, mace, and several others. All organisms showed the same pattern. This was important for the discovery of the structure of DNA, which came somewhat later. That's all for now. See you next time.